Okay, so proteins are huge blobs of something. They're hanging around in the cell cytoplasm. They have very specific functions, such as they can carry around things. They can help chemical reactions to happen quicker or more efficiently. In that case, they're called enzymes. So they can carry around phosphate groups, for instance. There's like a phosphate attached to a protein, and there is like another protein, and this protein maybe wants a phosphate, and the scope of this protein is just like to go around and leave the phosphate to others, or like carry around other proteins. And yeah, it really depends. They can do a lot of different things, but like the point is that they're really complex and they're really important. The DNA codes for proteins, most of the DNA codes for proteins, and um, living organisms are able to make their own proteins. Whatever can't make their own proteins is not even considered to be alive, such as viruses. They can't make their own proteins. So, how are proteins made? Proteins are made in protein synthesis. It's a process divided into two: transcription and translation. So, during transcription, DNA is being copied to mRNA. DNA is this double-stranded um, series of nucleotides. Those are just nucleotides, so they're double-stranded, and it's being copied. The information is being copied into mRNA. So it's literally the same information contained in the DNA. It's the same, but it's just less redundant. It's just in one string, in one strand. And so the idea is that you want DNA to be quite stable because you don't want to mess with it. If it gets broken, then there are processes to repair the DNA, but generally speaking, you don't want to touch it too much. So if you want to uh, access the information stored in the DNA, you want to read the DNA and then make a copy of it and then work with the copy. And messenger RNA is this copy. This copy is being made by the RNA polymerase, which is another protein. Notice that the RNA polymerase does not break the DNA. There is like this strand of DNA coming in, and then the polymerase just like reads it, like, you know, and then it opens it. Inside of it, it opens the double helix, doesn't break it, and then at the end, the DNA is still intact. So RNA polymerase just like going through the DNA strand, opens it, reads it, stores the information read inside one single strand, which is messenger RNA, which is this one, and then just closes it and leaves it there. So once we have messenger RNA created from RNA polymerase containing the information from the DNA, it's a copy. This messenger RNA goes into the ribosome. It literally attaches to it like cluck. And then the ribosome zips on it, let's say, reads as well. And then it creates this strand of, it's another strand of something. And the strand of something is called peptide. So this peptide is basically a strand of amino acids. Like messenger RNA is a strand of oligonucleotides, so just nucleic acids. A peptide is a strand of amino acids. Now, amino acids can be quite complex. Life uses 21 or 22 of them, and generally all amino acids must have this configuration where they have like this amino group, which is shown here, say transparent. So this is the amino group, and it's made by a nitrogen and two hydrogens, and there is this carboxylic acid. And so amino, carboxylic acid, amino acid. All amino acids have this structure. Then they differ based on what you attach to this side. You see that it's open. So if you attach, for instance, another hydrogen in this side, you're going to have glycine, which is the simplest amino acid ever that you can make. So the proper orientation will be this. So this is the oxygen, this is this hydroxyl group, 2 carbons, and then this amino group here. So glycine is the simplest amino acid you can have, but there are different ones. For instance, this is just a few. You can have alanine. It's when there is another carbon attached to it instead of a hydrogen. A valine, it's when you have a carbon and 2 other carbons attached to it. Isoleucine, leucine, and so on, methionine. They can have sulfur. So this is called side chain. The side chain determines which amino acid we're talking about, but in this case, all amino acids have this head. Okay, so the ribosome basically reads the information from the messenger RNA, just reads it, and it knows which kind of amino acids it should put together to create this long strand that we call peptide. So this is a better view of how ribosome works. mRNA strand goes in the ribosome, and the ribosome takes out like a peptide. But the point is that once you have a peptide by itself, like a long string of amino acids, they're not really functional. They're pretty much useless. You don't do much with them. You have to fold them. And the Golgi apparatus folds the peptide. So there is this peptide which is just like going around the cytoplasm, goes to the Golgi apparatus, and those peptides are probably also carried around by other proteins into the Golgi apparatus, which is a cell structure, and it's like an organelle, and it will look like this. So this is like a single peptide, and if you put more peptides all together, you're gonna have like a blob of those peptides, and they can be arranged in different shapes. They can be like this, or they can be like this, and the shape of those peptides will determine how the protein can operate. So for instance, in this area there can be like another protein that can bind to it, because it will find, for instance, 3 or 4 amino acids which it can bind to. So it's important that in this area those amino acids are present, so this protein can bind to this other protein, and that's literally it. And you can put more proteins together. You can do protein complexes. Like this is another protein. Just put it together, and then you have a protein complex. And those are different subunits, and you can have more of those. Then you can have like, yeah, ideally just infinite, but at one point it becomes just too complex and too unstable. But yeah, that's pretty much it. And all this blob that we see, it's just like this protein blob. So proteins are being made into steps during transcription and translation. In transcription, the DNA is being read by RNA polymerase and copied. The information is being copied in messenger RNA, which then is being processed by the ribosome, which creates a peptide. So a long chain of amino acids. A way to remember what is transcription and what is translation is that during transcription, the information from the DNA, which is stored in nucleotides, it's still stored in nucleotides. So the information is just transcribed. Same language, just being transcribed. During translation, the information is being transcribed from nucleotides to amino acids. And so it's like another language. So you have to translate it. You have to translate the nucleic acid language to the amino acids language. So transcription copies the information using the same language, nucleic acids. Both DNA and RNA are nucleic acids, just long strings of it. And translation changes language from nucleic acids to amino acids, forming peptides. And more peptides all together will form, like they're called like subunits, will form different proteins. And proteins can have different functions depending on their subunits, depending how they are arranged, depending how big they are. And yeah, it's pretty complex topic, but those are proteins in a nutshell.